Run.
The Bostonians who started Grey Thumb always had their meetings with places with lots of good beer, so they probably had a better insight than I do. But this is Grey Thumb, and it stands for sort of green thumb is a gardener of Terran biology. Grey Thumb sort of gardener of technology. The products of they came up with this several years ago, and they formed it as a homebrew computer club for artificial life experimenters, practitioners. So if you know what the Homebrew Computer Club was, it was this group of geeks that met in the garage, and then they met at Stanford starting in 1975, and they brought in their Altair kit computers and they tore them apart and they showed us stuff off, and a couple of the kids were Steve Jobs and Steve Wozniak that were showing off what they built, and so it was a sharing kind of forum, and out of the Homebrew Club came the whole computer industry. Chunk of it came out of it, microcomputers. And so I don't know if huge industries are going to come out of this, but certainly a lot more creativity can be had if you have a social group to meet with, especially around a topic as strange as this one. My interest and background in this is I've built some kind of artificial life-like systems in the mid-'80s when I was a graduate student at USC. There wasn't a field of artificial life, so they were called cellular automata, and I built them on VAX computers connected to the ARPANET, or the internet at the time. And then in 1996, I formed an organization called Biota, which is a subset of another organization I formed called the Contact Consortium, which does all this stuff to do with avatars and virtual worlds. And I always felt that inside virtual worlds might be in a chance for a rich enough little ecosystem for artificial life experiments to happen. So that's why biota.org was formed. And we had 4 conferences starting at the Burgess Shale in 1997 and Cambridge, England. It was quite a great conference series. Really interesting people like Richard Dawkins involved in it. And then Tom Barbalet, about 2-3 years ago, started our podcast. And the podcast is sort of the modern way to do a conference, is to bring in thousands more people. And we podcast initially all our old conference presentations, and then we went to interviews, and now we have a live radio show once a week that you can call into on the regular phone, and it's recorded, and it's called Blog Talk Radio. And it's been a lively, amazing run of great conversations of historic figures like Steve Grant who come back into the fold, to new projects, new people, new visions, paleontologists calling in, immunologists, people from different fields that have a passion for this idea. So that's where Biota is. And so back in March, Tom and I thought it was time to go and talk to Grey Thumb. So I went out and did a meet and greet and had lifted some beers with them on March 3rd, and then we established a really good friendship partnership with the Grey Thumb, many of whom had been listening to the podcast. And I suggested we start to start chapters. So I said, well, let's start a Silicon Valley chapter. They started a London chapter, and now there's a chapter in, I think, starting in Brighton. To this day, June 24th, is establishing a first chapter meeting in Holland. So it's spreading. Whatever it is, it's spreading. And one of my personal interests in all this is the PhD proposal, the work that I started at USC in 1985. I always wanted to get back to it. And I was offered a position at a university in London as an external graduate student to do a PhD, or a practice-based PhD, about a year ago. And then I basically decided to go for it back in January, wrote up the proposal, and it's actually a proposal to continue the work I started in 1985. And my old professor from USC wrote a letter to the university so that I could be classified as a continuing PhD student, which is sort of funny with a 22-year interval. It's a large, but I showed that there was a body of work in between, and mainly was that processors improved and it was possible to do the kind of thing that I was thinking of. And that work particularly has morphed into an idea that we as artificial life practitioners can't have a healthy medium if we build things in isolation. If we put all your energy to build a little walled garden experimental universe, you'll run out of energy or resources or daytime hours, nighttime hours to continue it after a while. If these systems were connected into a larger grid, where even though they're different, they have different properties, objects can be sent back and forth between the environments. And objects might be creatures or just stuff that is flowing, stuff with APIs and affordances that goes from one world to the other in kind of an encapsulation, and then instantiates in the new world and does what it tries to do. That will unite our efforts to create, to create evolutionary systems in ways we can't even predict. You'll have that fantastic, weird side effect of look at all the weird stuff that's happening that we could never predict. So I came up with a name for this vision, which is the Evo grid or evolution grid. Why grid? Well, evolution's obvious, but grid computing is sort of the basis now for a huge amount of what's done in science and in multiplayer gaming. So, Second Life is basically a huge grid of machines with these portal viewers called the Second Life browser that's looking at different segments of the grid. World of Warcraft's the same way, and then there's gene grids, and SETI at Home was one of the original grid systems. So if we had an artificial life grid or an evolution grid, we might be able to really achieve something as a group, and all our A-Life boats would rise along with this grid. So that's actually was the proposal for the PhD work, and they accepted it, maybe because they didn't understand it. So I had no risk of them rejecting it. And I'm going to recruit a great community of people. One of them may be Richard Dawkins, because he spoke at our second conference, and I went to have tea with him. I can't have tea with him anymore, probably, but in Oxford about 5 years ago, and described this vision, and he really liked it and wanted to be part of it. So I'm going to go try to have tea with him again. You can't get on his calendar anymore, but and see if he would want to be an advisor to the project or somehow associated. And we'll get all the original names of people that have, Carl Sims and all the original folks, Tom Ray, like that, could be part of it too. And so there's one more little piece of the puzzle that's come in, which is talking with people like Richard Gordon. Richard Gordon is an immunologist in Manitoba in Canada. He was at Digital Burgess, which was our first conference where we climbed to the Burgess Shale up in the Canadian Rockies. And he's an amazing guy. And he and another professor are putting a book together, which is going to be published in October by World Scientific in Singapore. And it's about 25 contributing authors on questions of God versus science, God, creationism, intelligent design, evolution. And it's got chapters written by philosophers, by a cardinal, rabbis, evangelical thinkers, biologists, etc. The head of the Vatican Observatory has a chapter in there. So it's going to be quite a book. It's 900 pages, so it's not the kind of bedtime reading, but it's going to be coming out. And he's whenever I present the Evo Grid idea to him, and this is a concept for you guys to really think about. He said, how do you know that you have real evolution going on if you have all this artifice, if you have created a structure of genes already? You said, well, let's create a body and create a phenotype-genotype relationship and have a creature made. That's kind of, you're already sort of halfway there. You're already putting a lot of invention into it. Is it working? Oh, there's still...
Not yet.
Okay. I'll keep talking. Maybe it'll work off that machine. So, just to finish the idea, he was challenging the assumption. He's saying, really, when it comes down to it, and his book chapter is called the artificial origin of life. He's talking about artificial life, saying the only way you can be kind of have the litmus test, pass the litmus test of whether you have a system that is exhibiting real properties of biology, is if you start from scratch. If you start from, there's Fred Hoyle wrote something in the 70s or 80s that talked about having a tornado go through a junkyard of parts and suddenly the 747's assembled. Well, sort of taking off on that challenge, starting with the origin of life problem, a jumble of chemicals, and if you run a chemical soup long enough, you get vacuoles form with bilipid layers, you get stuff inside that can repair the bilipid layers, and you get a blueprint that can be used to copy the vacuoles, et cetera, et cetera. Sort of, this is something thought about in the origin of life community all the time. But what if you created an arbitrary set of statistical simulations and ran them with large grid or multi-core processors, and could show that replicators would emerge within that and persist and develop the properties we might call lifelike? Richard thinks that is a much more grander challenge that our field could take on. It's that really low level from low. What does it tell you? It tells you that yes, in principle, this could happen in a simulation, and if you could create enough, is it hard or easy? It casts some light on the origins of life a little bit, but it will teach us a lot about how hard it is, about how we might look at it in chemistry, because we may not be trying to simulate the chemical world because we can't. It's too much computation to do that. This is just ridiculously computationally intensive to simulate normal chemical interactions. But it might be that that's kind of a good crisp thing that we could get our teeth around as a community, is to do that origin of artificial life, stepping really back really far. Now, of course, what are the starting parameters? So if you're an artificial god, and you're playing, we may have found a solution.
We're ready to go.
Turning 90 degrees, we find a solution. So the last thing is, so you'd say, well, you're just an artificial god, you're just setting the starting parameters for this soup, and of course things come in. And it's at the center of debates over religion and creation and origins and things like that. It's just such an interesting, it's an open-ended, interesting project that one could take on. And it may be that it's a big science project. It's maybe not a hobbyist project, maybe it's thousands of multi-node computers and some wacky person who tries to get funding for this and keep it going, and then one day, or do you need to recruit 100 million home computers, and the one that generates a replicator, you could win some prize. It's like SETI at home, first person to find a signal. Anyway, but that, I'm rambling on here, but that's the idea. Is it's something to talk about later, but do you go for this really fundamental grand challenge, or do you stick with building things that are lifelike and see if evolutionary properties appear in them and maybe try to grid them together? So, and I'm heading to London in 2 weeks from Saturday, a week from Saturday, to start this whole crazy enterprise. And that's my report. Take it away, John. John Cumbers, Brown University. Prior to that, in the UK, presumably.
Right.
Somewhere.
London.
And then last 3 months at NASA Ames.
I've been at Ames for a month, and I'll be here for 3 years or more, doing research on synthetic biology and how it will apply to space settlement, biological ISRU, and ISRU is in situ resource utilization. So in situ, where you are, resource utilization, utilizing the resources for manned missions in space. And in space, we mean an asteroid, primarily the Mars or the Moon at the moment. 5 years ago, I was a software engineer. 3 years ago, I was a bioinformatician. Now I'm a biological engineer. So I know where you're coming from if you come from the software world. And I'm hoping to keep everyone with me when I talk about what we are doing with biological systems and how we're hoping that we can treat them the way that we've been treating electrical systems and computer systems, in that we are now at a situation where you can fairly easily program a computer to do whatever you want it to do. We're only just at the beginning stages of programming biological systems so that you can do what you want them to do. The talk is interwoven with cool pictures of space, inspiring you of what we're going to be able to do there, with some fairly simple biological systems that we've managed to control. And I'll show you some of those examples. So this is the grand vision, that whenever we travel to another place in the solar system, at the moment the mentality is that you take everything that you need with you for the trip. And that's got to change. That mentality's got to change to one where you don't take it, but you make it. And why is that going to be a possibility? We know it's going to be a possibility because if you look all around us on Earth, most of the building materials, maybe not most of them because stone isn't, but most of the wooden building materials, most of the things that we see around us, human beings, everything is made out of carbon. It's made out of carbon dioxide that used to be in the air. The carbon is now fixed into things that we can see around us. So there's no reason why that couldn't happen wherever we go, that there's a carbon source. The carbon dioxide content of the Earth is negligible, it's very small, less than 1%. On Mars, the carbon dioxide content is 97%. So there's a big problem at the moment in that we're putting too much carbon dioxide into the air from burning fossil fuels here. If we went to Mars, there's a huge amount of carbon dioxide in the air, and I'm really happy about that because I'm going to build things from that carbon dioxide that's there. So what you can see is a schematic of a photobioreactor, which is a bioreactor, which just means a place where cells can grow, and a photobioreactor meaning that the input to that system is light, and the output is whatever you want it to be.
Sounds like an aquarium.
Yeah,
Maybe
I guess, yeah.
John, if you're not going to mention the fact that the Earth's atmosphere is a bioformed thing from algal mats and stromatolites.
Sure. So the current theories on where what the Earth's environment was like before and what it's like now, which is it was before lots of carbon dioxide like the Martian atmosphere, and it is now 23% oxygen, 70-odd% nitrogen. And the reason being that there's a lot of carbon dioxide, the current theory is because of all the explosions during the formation of the Earth and during the formation of planets that throws out a huge amount of carbon dioxide. And it's small, single-celled, photosynthetic organisms that have taken the water, split it, and released the oxygen into the environment, which is now one multicellular life like us couldn't survive because we need that oxygen in the environment. There's work in NASA going on about that it may not be single-celled algae that did it. It could be a lot to do with compression of different rocks that produce the release of oxygen, which I was just learning about recently and didn't know about. So I'll speed up because I wasn't planning to talk so much and whiz through the slides. And please interrupt as we go, particularly if I've said something that I've taken for granted and isn't granted, because I don't know this audience. So I like to set out a goal. I think Kennedy did this very well at the beginning of the 60s. He was talking about sending a man to the moon and back again. And he said, I believe we possess all the resources and talents necessary, but the facts of the matter are that we have never made the national decisions or marshaled the national resources required for such leadership. We have never specified long-range goals on an urgent time schedule or managed our resources and our time so as to ensure their fulfillment. If you ask anyone at NASA at the moment about getting any project done, big project, it generally can't be done we're not talking in a decade, nearly anything that we talk about. I've been there a month, but that's... that seems to be the way that we're talking. But Kennedy in 61 said to Congress, we're going to do this. And I think in 62 he said, and we're going to do it by the end of the decade. And I think that's what we need. If we're going to take the next step and we're going to be living elsewhere, we need the political will to say, let's do it, and let's do it by this date, and let's do it by this, in this budget. I think that's the only thing that's stopping us. I don't think we lack any of the technical ability to do it.
Are you talking about living elsewhere?
I'm talking about a stable colony or stable settlement off planet.
On Mars?
On Mars, on an asteroid, on the moon.
Do you mean a self-sustaining?
Yeah. I mean everything. Like, yeah. And yes, it's
populated with humans.
Yeah.
Okay.
Yeah. I mean, and I'm throwing this out there as my vision, and Kennedy threw it out there as his vision. And if you speak to people about the history of the Apollo program, no one was really sure whether it was going to be possible. They could do it, but that's not the point. That's the point of a vision, is to try and predict something that you think could easily happen and you want to happen. So I'm throwing that out there as that's my vision, what I'd like to see. And I think that we just need to plan and do it and spend the money. So, and the reasons why we might want to do it are multiple reasons. One, for spreading humanity. Two, that something's very likely going to hit us at some point in time. We could talk about the philosophy of the destiny of humankind.
But you'd like to live on Mars?
When we've got it to a state where it's an environment that you want to live on, then yeah, sure. I'm an explorer. I love to travel, so yeah, that's what I like to do. My wife would not.
You can do Chinese?
So I'm going to. The talk is an outline of this vision for space, the vision for how we're going to do it, which is by making biology easier to engineer, some applications of synthetic biology, and then I'm hoping that our discussions will be able to fit it into the AI context. And I think you'll see from where we are programming simple genetic circuits in organisms, I think you'll see the potential for applying this to your field. So this is my definition of synthetic biology, making biology easier to engineer. And we'll talk about why it's new and why it's different in a second. This is a quote by a quote. This was something that Drew Endy wrote, who's a professor at MIT at the moment. He's moving to Stanford in the fall. He said, in 1978, Cybulski and Scala wrote, "The work on restriction endonucleases, which are enzymes that cut up DNA, not only permits us easily to construct recombinant DNA, meaning shuffle genes around, make changes to DNA, and to analyze individual genes, but it has also led us into the new era of synthetic biology, where not only existing genes are described and analyzed, but also new gene arrangements can be constructed and evaluated." So this was in 1978 that they wrote this commentary saying, wow, this is big. Scientists have shown that you can chop genes around, you can change bits. This is like a whole new world of synthetic biology that we're entering. But when you look at what's happened 27 years later, this is what Drew writes: despite tremendous individual successes in genetic engineering and biotechnology, why is the engineering of useful synthetic biological systems still an expensive, unreliable, and ad hoc research process? So we've been able to do it for 30 years. Then why is it that it's still incredibly expensive to design a genetic circuit, incredibly difficult to make it work, and requires a huge amount of skill in order to do it? And there are two camps. One is biology is very, very complex, and we're not going to be able to engineer biological systems as we engineer computer systems or electrical systems. And the other camp, which Drew is in and I'm in, says it's difficult because we've never made it easy. And that's what synthetic biology is about. It's not about doing one-off ad hoc applications, producing a protein here or making a drug there or making a system that does that. It's about foundational technologies that makes it easy for everybody to do that. So that when you've done something, designing a biological system, you've passed on common tools that other people can then use to make it easy to engineer the systems that they're working with.
Is that because of lack of funding and lack of support?
No, it's because, I think it's because it's been science, and science doesn't work in the same way that engineering works.
So we should have something, articles like 7 ways to hack your plants?
If you saw Make magazine
Yeah.
from last year, did you see it?
Yeah, I did,
Yeah.
Yeah. So that's taking it to the people, so to speak. right?
Yeah, which is a whole nother talk on DIY bio and how you can start doing cool biological experiments in your kitchen, which then opens another can of worms of should you be doing cool biological experiments in your kitchen. There's a bar camp happening in August at Institute for the Future in Palo Alto that I'm part of, called Bio Bar Camp. And there'll be
Yeah, I'm going to that.
Oh, are you?
Yeah.
Are you on the mailing list as well?
No, my friend Jimmy Cascio is, and another friend of mine up in Seattle. Some people are involved in it.
Okay,
so
great.
I only know his Twitter. I actually can't put a name because I only know him through Twitter for so
Okay.
long. I can't remember his real name, but he's organizing it. And I promised a couple people I'd go. Yeah, so I'll be
What
there.
are the dates and what's happening at it?
It's August 6th and 7th. The 6th, we're hoping, in the morning is going to be held, although we haven't contacted them yet, but we're hoping it's going to be held at Tech Shop in Menlo Park.
Cool.
And we're going to do a DIY bio there, and hopefully get Tech Shop interested in doing it. Then it's a bar camp, so who knows what's going to happen, but it's like 120 people starting, then the main session starts midday on the Wednesday the 6th, and then introductions and session planning, and then a meal in the evening, and then the full sessions are on the Thursday. And so the bar camp, the idea is that people come and write up a topic that they're interested in talking about, and other people write their name below if they're interested in signing up for that session, and so it's just evolves. It's an unconference.
Like Bill conference.
Yeah, exactly. Yeah. So yeah, that's something that's going to be very interesting, and that sort of combines. That's not just synthetic biology. It's not just DIY bio. It's personal genomics and biotechnology. A whole load of stuff going on. Okay, so here's a genetic switch. What do I mean by genetic switch? I mean a way that genes are regulated. So here you can see a piece of DNA. Can people see that, or would you like me to turn it down? Is that better? Okay. Okay. Is that too far, or is that okay?
That was the perfect angle for me.
So here you can see a piece of double-stranded DNA, which you've probably seen before, and the big blue thing is a protein that's bound to it. And I have a question that I'd like to ask, if I could tell you how a gene relates to a protein. So I said it would be an interactive session. I hope that I could have some clickers so it was anonymous, but let's try it anyway. So I'm going to ask A, B, C, or D. So A is yes, I know the answer. B, I think I know the answer. C, I have no idea how a gene relates to a protein. And D, genes have no relation to proteins. So, okay, any for A, yes, I know the answer. Okay, any for B, I think I know the answer. Okay, any for C, I have no idea how a gene relates to a protein. And D, genes don't relate to proteins.
Just kidding.
So before I changed from a software engineer to a bioinformatician and a biologist, I couldn't tell you how a gene related to a protein. And if you ask people on the street, people generally can't tell you how a gene relates to a protein.
Or what they are.
Or what they are. And now that I understand what a gene is, what a protein is, and how they relate to each other, I'm just so excited to tell people when I sit next to them on a plane, when I'm like, right, okay, we've got 3 hours, right? which normally ends up with people on the edge of their seats by the end of the, not trying to leave, but because I think it is the most amazing thing, molecular biology, exactly what's going on in every single cell of our body, which is a complete copy of all the genes that regulate from a single fertilized egg 9 months later to a complete human being, all in one copy or two copies of your genome in one single fertilized egg. And as that egg divides, 9 months later, you've got 100 trillion copies of the genome, one double copy in every single cell. And in every single cell, you've got things like this going on, which is turning different genes on and different genes off. So that how do my brain cells know to become brain cells? How do feet cells know to become cells that are going to be in my feet or produce feet? It's all through genetic regulation, and it's all through turning on different combinations of those 20 or 30,000 genes to make different combinations of proteins. DNA is converted into RNA, which is a messenger molecule, and RNA is then translated into proteins. So there's a whole mechanism that comes along and can read DNA, can duplicate it, but can also turn it into RNA, and then the RNA is read and can be turned into proteins. And there's some really cool videos that I haven't got time to show. So the thing that we're doing in synthetic biology a lot of the time is regulating when these genes are turned on and off. And the body's really good at that. It's good at doing that to produce all the different proteins that are in your body. And a lot of your body is made of proteins. They're the things that do things. They're the enzymes that change the chemical reactions of life. And recent advances have shown that we can pull genes out, we can swap them around, we can take the human gene for insulin, we can chop it out of a cell that it's in, we can put it in a bacterial cell as a single cell, and we can trick the bacteria into thinking that the insulin gene is the bacteria's own genome, and it starts to produce starts to read it, and it starts to produce the insulin that's on that gene. We can then take that insulin and we can give it to diabetics who are in need of insulin. So it shows the utility and the common sort of place of genetic material in the world, which is pretty incredible. Recently you've heard about the sequencing of the human genome. That's reading the entire copy, single copy of one human person. And you've probably heard about personal genomics, which means that it's now just a few, maybe 100,000, probably cheaper to sequence the majority of your own genome if you wanted to do that. And you would definitely want to do that at the moment. I'd quite like to do it. It's just prohibitively expensive for a graduate student to do. But you might want to do that to see whether you have a particular version of one gene or the other, and that's the field of genetics.
A funny story, there's a professor who's an African American. He's a professor of African American studies, and he had his genome done, and to his horror, and he found that he had virtually no African genes. And he said, am I under false pretenses? Do I go back to my faculty and say, I'm not African American, I'm actually Irish and this and that. Anyway.
So I think there's a big lawsuit at the moment in California because there's companies like 23andMe, which you can give them a cheek swab, and they'll sequence particular parts of your genome and give you information about it. And there's a lawsuit from physicians saying that it should be only a physician that's giving you that information. It's going to be interesting to see how that pans out. So you've heard about sequencing, which was a huge deal to sequence the human genome. It cost billions of dollars. Now it's very, very cheap. You could get a read of a particular sequence of DNA for $3.50. Not the entire genome. That would still cost you $100,000 or more. But it's not billions of dollars that it was before. And it's following an inverse Moore's law. So every couple of years, the price of sequencing is halving. So it's very, very cheap now. Similarly, and this is a graph from The Economist, you can see the cost of synthesizing DNA is also following that same trend, that every couple of years it's halving. So at the moment it will cost you 69 cents a base pair to synthesize a single A, C, T, or G. It's halving every couple of years. So although at the moment you could pay $100,000 or a few $100,000 to literally synthesize an entire genome, which is prohibitively expensive, in a few years it's going to just cost a few $1,000s.
What do you get for $1,000 at 23andMe?
Well, they're a sequencing company, so they're reading it.
Oh.
But I'm talking about writing it.
Writing it.
Yeah. What would $1,000 buy you for 23andMe?
At this point in time, yeah.
I think their package is under $1,000 at the moment.
Huh?
But it's not an entire sequencing of the genome. But basically, you don't need to know the entire sequence of the genome to get some interesting medical information from it, because 99.9% of all of our DNA is the same. But I might be susceptible to heart disease and you might not. So we need to find the differences between people who are susceptible to heart disease and people who are not, and give advice to me to exercise more, and advice to you to stop smoking, maybe if you're susceptible to cancer. So you don't need to read it all, you just need to read the bits that matter.
Yeah.
And of course the bits that matter, that's what the field of molecular biology is doing, is trying to find those bits that matter, and it's not an easy process, because evolution hasn't designed organisms to make it easy for us to understand how they work. We're trying to reverse that. Now that you can write DNA very easily, we're trying to write genetic circuits so that they are well understood, and so that they are human readable, human understandable, so that we can engineer organisms to do what we want rather than just follow a random path of evolution. So here's the genome, here's a bacterial cell, DNA inside of it, and here's a piece cut out of it. This is what we've been able to do for 30 years. The analogy is that it's like a piece of software. If you can write the entire code, that's the Windows operating system, that's Linux running on it, but you can write whatever code you want and put it inside the operating system. Genomes are not designed like code in an operating system, but it's just an analogy that I think will get us fairly far. GenomeCAD, this is a great website where you can go and you can design genetic circuits. So I can't show you a demo, but you can click and drag, and you can say, I want this piece of DNA, I want that piece of DNA, put them all together, and eventually you'll be able to click a button, send it to the synthesis company, and they'll send you back the code that you've written, but in DNA. So you can actually get it in a tube. This is huge because you've been able to do it for a while, but with the falling costs, you're really going to be able to design on a computer, press the button and have it delivered the next day.
Yeah, it's a tech shop kind of thing.
Exactly, yeah. Okay, so here's one of the early transistor, and this is the parody that a lot of people make, that this is the next technological revolution that we're going to be coming to. The miniaturization of transistors has allowed us to have microprocessors capable of executing millions of instructions a second. This is where synthetic biology is going. That if we can take one of these circuits, one of these genes, here you can see a small molecule that's being added to the cell. It's influencing the way this protein touches the DNA, and it's allowing this gene corresponding this piece of DNA to be turned on or to be turned off. So it's a simple molecular switch. You add the concentration, it changes the conformation of the protein, changes the shape, and turns the gene on or off. So it's a basic switch. We want to see a data sheet like this available for genetic switches. So this is a data sheet that you might get from a transistor. You look it up in a catalog, you say, I want one of these that will work in this circuit.
It's like the homebrew people did. They were building them from the basic IEEE color code transistors. Yeah.
Exactly.
Yeah.
So if we want to achieve grand things, which is to be able to engineer an organism so that it can survive the harsh temperatures of the Martian environment. It has a mechanism to deal with the radiation of the Mars atmosphere, and it can produce a biofuel so that when I land on Mars, I have enough fuel to get back again, or enough glucose so that I have a supply of food, or enough oxygen so that I have a supply of air to breathe. We can design organisms for each one of these applications. If we want to be able to do that, then we need to start with foundational technology. So at the moment we're at this level, where we can design very basic things. We can chop, that's a bunch of stones, where we can chop out genes, pieces of DNA, put them together again, see what they do, tweak something here, tweak something there. We're not going to be able to get to the level of a sophisticated bridge by piling together lumps of stone like this. What we need to do is some sort of standardization. As I said, evolution hasn't given us standard parts, but that doesn't mean that we can't now take our ability to synthesize DNA and create standardized parts and standardized systems from it. And here's the wonder of biology. I'll let you look at this before I tell you what it is, but some people have probably seen what it is before. But we'll go in reverse order. So this is ivory from tusks. Just think about how hard ivory is, how hard teeth are, or bone. This has all come from biological material. This has all come from carbon sources around us, or calcium sources in terms of teeth. Look at this pine cone, if you ever pick one up, particularly at this time of year, and you look at it. One, it's incredibly beautiful. Two, it's incredibly manufactured. But three, it's so hard. It's, wood is one of the hardest things that biology can do. If you can think about going to anywhere in the solar system, you don't want to be taking building materials with you. We know that biology is very, very good at doing building materials, so this is, I think, one of the key applications of it. And this, these are the spinnerets of a spider. And so the purple is spider silk being produced from the spinnerets.
Is that artificial color?
It's artificial color, yeah. And there are people now who've put these into salmonella, which is another bacteria, and have got them producing the monomers from spider silk, which are the bits that form together. I'm not sure if they haven't got them to spin the silk yet, but they've got them to produce spider silk. And if you think how thick or how strong spider silk is, if you walk into a web, so that's a really, really useful building material. So the dream is that we can engineer these things and we can make these things, but at the moment we can't. Here's something that I'm working on at NASA. It's called Lingbia. You might have seen it. This is down in Baja in Mexico, and it's a photosynthetic bacteria that is colored green so that it reflects the sun, so that it absorbs the sun's energy and converts that and uses that energy to fix carbon around it. It makes these big mats of thick biomass, and it lives in high salt environments. So this is something that we're studying for its possibility as biomass for building, biofuel, and just its general use as a photosynthetic organism if you take it elsewhere in the solar system. Okay, so I've showed you how great how great nature is, how great biology is. Here's some examples of where we're at the moment in terms of synthetic biology, in terms of making biology easier to engineer. So this is Wendell Lim. He's a professor at UCSF here in the city. MAT-K signal cascade is a way that cells can communicate with inside a cell, how you get signals inside to the nucleus, which is where all the DNA is stored. And it's very ubiquitous. It's used throughout biology as a signaling mechanism for many different things that the cell wants to do. And what he's done is shown that you can tune the ability of this signaling cascade, or you can tune the output of this signaling cascade. And this was published in the journal Science a couple of months ago. So remember that I said that DNA folds up and makes proteins, and it's the proteins that do stuff. Here are 3 proteins, these circles. Here's a 4th protein, which is called the scaffold protein, which these other 3 proteins stick to. And here's a diagram of the cell. Remember, our body's made of 100 trillion cells, and there's a nucleus inside. All the DNA is in here. And if you want to get a signal from the outside the nucleus into the DNA, here's this signaling cascade is how you might do it. And it works by the protein passing on a signaling modification from one of these balls to another, boom, 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 and then down into the DNA where it would turn on or off. In this case, it would turn on different genes. So here's what it would look like over time. Again, you turn on this protein signaling mechanism with a small molecule. It's a pheromone used. This was studied in yeast, and a pheromone is a small signaling molecule. And over time, you can see the output increases. So as you give the signaling molecule here, it's off, it turns on, and then it levels out. That's the natural response of this pathway in a natural system. So what Wendell did was to take this scaffold protein and to stick another part of a protein on the end here, and the other part is the red protein. So how do you do that? How do you go about sticking another part of protein on the end of another one? It's quite easy, because we know that DNA encodes for proteins. So if you change the DNA, then you can change what it makes. And so we know that you can change the DNA because I explained about how you can just write DNA, type it out on a computer. I'm not sure how they actually did this. There are slower methods to do it. So that's what you do. You design it, you think, okay, I'm going to take this piece of protein, which I know has a particular function. I'm going to insert it into here. I'm going to look at how it would have been made, look at the DNA. I'm going to put the DNA in this part here, and then I'm going to make it. So that's what he did, or his graduate student did, rather. And he made it so that that red protein that he attached on, he knew it had a high affinity, meaning that it liked to stick to this blue protein here. And this blue protein he put as the output of this signaling cascade. So I told you that the signal comes down, 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 and then turns on genes at the bottom. The gene that is turning on is this gene, and this gene is going to bind to this scaffold, which he's modified. So he's modified it so that this has a binding region for a blue protein, and he's made it that the output of the signaling pathway is this blue protein itself, which is going to bind there. And why does he want that to bind there? He wants that to bind because it's known that this blue protein can positively affect the amount of signal that's coming through here. So it's a positive feedback loop. When this binds here, it's going to bind there. And what you can see, here's WT, which stands for wild type, which is what normally happens, which I just showed you. With this particular design circuit, he's got an increased positive feedback. So you've got an increased amount coming through. So that's one example. Negative feedback. He did a very similar thing, but he put in a negative regulator as the protein that binds to the end. And you can see here the output then is less than wild type, so it's reduced. A pulse generator. He did some other more complicated things of putting in inhibitor peptides that would bind and inhibit two other things binding. And you get another signal output, which is a peak. Another one where he turns it into a sharper switch, so that it's off and then on and then stays on.
Yeah, bioelectronics.
Exactly. So that's great. That's what I wanted to get across, that this is really good examples of foundational research. It's not a one-off application. It's not an ad hoc process. He's really engineering this signal cascade to give different human readable, human understandable inputs and outputs to the system. And you give it to an electronic engineer, and then they can start to think about the ways biological signals are processed and what they might be able to do in terms of programming biological function into cells.
Has this work been published recently, enough that they've got some excitement and some people working on it, or is it kind of in its infancy at this point?
This was published 2 months ago,
Okay.
this paper,
All right, so
Yeah.
a little new for.
Well,
in Science, yeah. So it was a big paper. And Wendell Lim was just announced as a Howard Hughes medical investigator, so that's a fairly big accolade in biological science. So
what was the title of the paper?
it was
Just a couple words.
using engineering scaffold interactions to reshape MAP kinase pathway signaling dynamics.
Okay.
It was published in Science.
Yeah,
Scaffold and okay, great, thanks.
yeah. And you can email me and I can send you this and I can send some other reviews. The paper's not that easy to understand because science papers are very short and they cram in a lot of information. So, so I think that's a great example. And the last thing, that there are some other things that they did in the paper that I didn't show, but one thing they did was to try and model, okay, how could we get any of these different types of signals out from a cell? And if they change the binding affinity of the protein, of the adaptive protein that they stuck onto the end, so they used a different strength binding of these two proteins. They changed the red protein to a different protein that didn't bind to the blue one as strongly. Could they get these different outputs? And they did. So they modeled it and then they showed that if they change it, they can get that as well. So this is one of the best examples of synthetic biology. It's foundational research, and you can see how it's showing the ability to control engineered biological systems in a rational broad sense. Okay, how long have I got, Bruce? For people.
When you were I think Jeffrey, how long was your?
5 after 8.
It's 5 after 8.
Yeah.
You probably.
Okay, I'll go pretty quick. So what's the difference between synthetic biology and genetic engineering? I think I've drummed that home. It's foundational technology. It's not one-off applications. It's broad things that are going to be applicable and make it easy for other people to engineer. And this is a favourite quote of mine from Tom Knight at MIT. The engineering of biology remains complex because we've never made it simple. He's holding in his hand a copy of the Registry of Standard Biological Parts. The Registry of Standard Biological Parts aims to do this for biological parts. So while we've got mechanical or engineered systems where we know you could go to a hardware store and buy a particular thread of screw and know that it's going to fit in the hole that you drill, you can't do that with biological parts, and that's what we're trying to do. So you can turn this into a data sheet like this. And a data sheet for biology for biological parts, they're stored in a database. You can look this up at MIT. If you want protein coding regions, terminators, regulators, all different genetic functions, you can click on one of these and you can pull it out. If you want to characterize it so that the other people can use it.
Has this been started already as a standard?
This exists for 4 years, I think, and there are 2,000 or 3,000 parts in it. Not all of them are physically available, and not all of them, the majority of them are not well characterized, so you can't just go and pick it up and know exactly how it's going to work. But it's the initial step and it's the first of its kind. But you can see here's another switch. You can see how it switches, what's the transfer function of input to output, the response time, things that you might want from an electrical switch. Here's some interesting stuff on the load of the cell. If you put your switch into it, how many amino acids does it take up per second? How many ribosomes does it use? A lot of this stuff is fairly difficult to find, fairly painstaking to do the experiments to get it. But this is the stuff that needs to be done if we're going to be able to engineer these systems easily, right? Because if we start to standardize around things like this, then we can really start to share information. We can start to share parts. and it's a foundation that we can build on. But it is fairly boring and fairly difficult to do.
What's hard? What's the hardest part about it?
I've not tried to measure these things myself, so I'm not sure. But certainly, it's not trivial to measure the number of ribosomes that your genetic switch or that your piece of DNA would attract per second.
There's still not a lot known about some of these.
This was just published, or it's about to be published in Nature Biotechnology, and I haven't fully read it, but I'll read it and let you know how difficult it is. Maybe it's not so difficult, but
So does your imagination leap from this foundational work to see sort of intuitively how to synthetically engineer organisms for, say, the Martian environment or the Martian poles, or?
definitely. Once you work at this foundational level, whether you're designing algae to live in the desert in Africa and take biomass and produce a biofuel, or whether you're designing a algae to live on an asteroid and be hardened to radiation and be able to produce building material. They're both engineering applications. It's just the framework that it's like whether you're designing a circuit board for a washing machine or a rocket. You could take a qualified electrical engineer and ask them to do both. It wouldn't be a problem, because it's all to do with regulating the genes inside the organism, and that's what this foundational technology is making it easy to do.
So I'm imagining that there's kind of a different discipline between, say, if you're a circuit board designer, you're designing the layout of a chip versus putting together gene sequences and sort of knowing how the proteins will fold. Do you see that's true?
Yeah, definitely. And what Drew Endy has proposed is an abstraction hierarchy, which means that for biological engineers, you have one person who's working on the level of the DNA. They might be interested in a particular protein, how the protein folds, how the enzyme changes, how the biochemistry works. Another person might be interested in a device, making an input-output system from a genetic circuit so that you can read polymerases per second, which is a common signal carrier that would allow you to design genetic circuits in the way that you design electrical circuits. Someone else might not be bothered with those two things, but they might be working on a higher level, which is making devices, how these different things connect together. So I think you're asking, well, electrical circuits are not the same as biological circuits, so...
Is there an intersection between the two, like, say, between synthetic organisms and micro-robotics?
Micro what, sorry?
Robotics?
I don't know. I'm not sure.
Presumably there's a whole class of problems you want to solve. I mean, if you want to solve the problems of doing chemistry on Mars, creating oxygen from ice, it's a different one than solving the problem of mobility, creating something that can move that's a megafauna. So it really depends on your design space. I'm going to solve this one little problem here. I want to land an empty habitat and have it full of consumables by the time the human crew lands.
Sure.
It would be design space as well boxed.
And a lot of people at the moment are just working on single-cell organisms. So there's one cell that does something, not engineering whole multicellular things to run around and jump through hoops.
How do mutations play into this?
So mutations are happening all the time, just through radiation or environment, or just random changes in DNA. So it does come into it. For the systems that we're talking about for space, I'd envisage that you'd have a frozen stock of your cells, and that every so often you'd re-inoculate, you'd reinfect the bioreactor that you're running the system in. So that because random mutation could happen and it could throw everything out, and your system could die, or it could mutate to something else, or it could just start doing something else.
That raises an interesting point, which I've been thinking about this whole time. And that is that it sounds like the kind of design that you're talking about is anti-evolutionary. It's not based on the same biological processes that all of this stuff is been doing for 4 billion years.
Right.
You're taking this stuff away from its evolutionary pattern, and you're taking it and you're putting it into a pure design space, an engineered space.
Right.
And so I'm wondering if, this might be a question for everyone else here too, is how does this relate to the question of evolutionary design versus rational design? And do you think, actually I'll just ask, do you think it's possible to create life on Mars without evolution?
I think it is possible, and one of the, but I think you probably want to use the tools of evolution. For example, if I wanted to design an enzyme if I have a system that can produce the enzymes to produce a biofuel on Earth, and I wanted to take that system and put it into an organism that could survive the temperature fluctuations on Mars, then I might use evolution to do that. I might take the individual genes in my Earth organism, I might use directed evolution or artificial evolution and decrease the temperature.
Vehicles are shipped in a sealed box to the Cape for launch, and it was always presumed to be clean of biological entities, but it turns out that over several years, there's some bacteria in there that are extremely well adapted to that ultra-dry environment that can survive a pump-down vacuum. And so we sort of created this wonderful little laboratory where we allowed nature to select or otherwise adapt. Just like in beer mats in Britain, somebody did a study of the organisms that live in beer mats, and there's quite a family of things that have evolved from beer coasters, and they go from beer coaster to beer coaster, and that's their universe. So the idea for your grid, if you think about it. That's right.
Exactly.
And there is life on Mars. It is contained inside the parts. Doesn't matter whether they're gas filled or not. There's bacteria all over the interiors of those spacecraft. And as soon as they're exposed to the radiation coming in to Mars, they're toast. I mean, they're not going to make it. But the inside spaces and the crawl spaces, and in between the wiring harnesses and in between the little wires and everything, they're all over the place. So, and they're happily just sitting there. They're not really metabolizing, but they're probably sitting there, slowly expiring.
So I will whiz through to the end. Some other stuff we're doing on trying to standardize things, some history of the way the ohm was standardized via somebody taking a measurement and setting a reference standard, and that's the way we're trying to measure genetic output at the moment. And I think that, so back to the vision, we're looking at an asteroid. There's plenty of biological material waiting to be made there. Different ways that we could do it.
Why asteroid and not the moon?
The asteroid, because if you can do it on an asteroid, you can do it anywhere. You can do it if you can develop technology for an asteroid. You could definitely then use it on the moon.
So why not start with the moon? Because it's easier.
Well, there's actually a short answer. The moon is actually, it's a harder place. It's a much harsher place. Think of it as an incredible desert because of the way possibly the moon was formed. But the asteroids, or near-Earth objects, are sometimes called, or icy objects, are the primordial material of the solar system. So they're well mixed, and there's a range of there's things that there's methane ice, there's water ice, there's all kinds of organics, metals, you name it.
So asteroid is easier.
It's easier. It's what we're made out of, because certainly after the cooling of the Earth, there was a lot of impactors that come in, gave us our oceans and everything. So we're kind of made out of that stuff. So by going to the asteroid, you're kind of going home. In a sense.
More raw material to work with.
Yeah.
As opposed to.
And it's easier to get to them.
Yeah,
Because you don't have a gravity well.
Yeah. Right.
And they're passing by all the time. Sort of stuff.
Convenient.
They're entering the atmosphere constantly.
Just quickly, you could synthesize. Craig Venter has shown that you can synthesize entire genomes. So you can go to a database on the internet, download the genome that was previously sequenced 10 years ago, send it to the company to synthesize it, and they can send you back big chunks of DNA. You can put those together. He's completed an entire genome of a bacterium. and I'm working on organisms that can survive extreme cold, and that's the main work that I'm doing. A lot of people in the Bay Area, Drew and Christina Schmolke moving to Stanford, Chris Anderson and Jay Kiesling at Berkeley, Adam Arkin at Berkeley as well, Wendell Lim and Chris Boyd. And biobricks.org is a foundation for making standardized biological parts that you might want to check out. And you can email me with any questions. My email's on the list there. So.
What's that name?
Biobricks.org.
And you've got a blog going, and
Yeah, I haven't. It's not updated. So it's a great.
and
Yeah.
this video will be on Allan's site and linked into biota.org
Right,
and
yeah.
Graython as well in a few days, or who knows when.
Pretty soon.
Pretty soon. Well, any more questions?
A quick one. The circuit you showed used solely protein.
Right.
I can imagine other things that go through have transcription as a part of it.
Mm.
It seems like a lot of the way biological cells work, differentiating is that.
So in terms of RNA, you mean like?
That a critical part of the functioning might be to modify the transcription rate of some other gene, so that the circuitry includes transcription as part of it.
So the ones that I showed you, the arrow in a couple of the ones towards the end, the blue protein that bound to the red part was produced via transcription.
Oh, okay. So it is modifying the
It
rate.
was feedback. Yeah,
Okay.
there was a feedback mechanism in it.
Yeah, I'm not familiar with biological systems, but 10 years ago when I've been told about it, I've been told also that one of the major problems is the expiration date of biological systems. Is it the current, can you build something from biological matter that can last aging? We talked about
Right.
aging. So what's the current problem that we are facing with such biological systems?
We're not yet facing those problems.
Not yet.
We can't yet engineer biological systems the way we'd like to. We're still at a very primitive stage of producing one product or one sort of cool thing that flashes or one thing.
And dies after.
I mean, yeah, there's a good example from Michael Elowitz at Caltech of an oscillator, and it uses green fluorescent protein and it flashes on and off and on and off and on and off over a period of hours. And it flashes about 8 to 10 times and then it fades away, it dampens.
So I guess that the one problem that I've noticed is getting the equilibrium. So when do you know that, how do you get this equilibrium of not dying but not exploding? So I think this is a major problem in such evolutionary systems. Also in Evergrid that been taught here. So how do you spread something and it gets into equilibrium with the surrounding environment?
I think that is a really difficult question because life tries to dominate all the resources that it can. If it doesn't die, first you try not to die and then you try and dominate all the resources. So I don't know. I don't know the answer to that. Certainly you can do it in a controlled environment, and that's what we do in the lab.
Yeah.
It's almost like we have to go through the DARPA Grand Challenge phase. The first DARPA Grand Challenge, Red Whitaker said, we ran the record-setting robot ran off the road after 11 kilometers. It just shows that after 50 years of autonomous robotics, we got nowhere. It was a reality test for the whole robotics community, and then they,
far away from where we want it to be.
Yeah.
So far, your focus is on inserting traits or building blocks systems. Are you doing systems that can self-replicate or self-organize? And I guess that would combine with your question,
Yeah.
because
Yeah.
once they can self-replicate, then they will be replicating according to the context that
Yeah.
they're in.
It can oscillate.
Be adaptive.
Sure. So these systems, all of them replicate. When we look at when I show you one protein that I showed you, there may be, I don't know how many millions of proteins of those proteins, probably hundreds of thousands of those proteins inside a single cell, and each side, and in the lab, when we deal with a Petri dish, you're talking about millions or billions of cells on a single Petri dish. So they are replicating the systems that we're talking about. They're already self-replicating. By definition of putting it into the genome, when the organism continues to do what it does, it
But
replicates.
that's not part of the interesting feature.
Yeah, it's again, at the moment we're not at the stage where we can get them to do what we want easily as a one-off, to yet think about using replication as an interesting feature. But it's something to think about in the future.
Has anyone thought about self-repairing systems?
Again, that's
similar thing.
very interesting and something to think about. But at the moment, we're not able to get a system that. Yeah, that's an interesting.
Yeah, self-healing systems is very.
It's difficult enough to get the system to work
Period.
in the first place. To think about trying to get it to repair itself when it doesn't work.
It sounds like a ground floor opportunity. And with that, I think we're gonna, Jeffrey has, I want to thank John for giving this nascent group a look into the very beginnings of the homebrew movement. And
Yeah, I'm happy to talk more.
And come down, and if you come down to see him at Ames, you're going to see something which I love. I sort of am a fan of algal mats, but they have this sort of cube, a piece of algal mat, and if you shine a spotlight on it, you can watch the oxygen content go up really quickly. I mean, just at Yuri's night.
Oh, cool.
Yeah, it was like, oh, it's climbing right away. So it's like this little generator. Just responds so quickly.
Who was that? Was that Sergey doing it?
I think so, yeah.
Okay, I'll ask her.
It's like, cool, it actually works.